So now we're going to learn about genetic mutations. First, let's define what a genetic mutation is. So mutations are alterations in the DNA sequence that change the normal code carried by DNA. So gene mutations can occur anywhere along the DNA molecule, and typically they're going to be by random chance, or they can happen because of an environmental factor. So what are some environmental causes of mutations? Well, X-rays, UV rays, cigarette smoking, lead poisoning, and pesticides. So when gene mutations occur, gene expression is often affected. Therefore, the cell assembles different proteins. Usually, the enzyme DNA polymerase repairs any mutations. However, the repair system cannot keep up when there's overwhelming numbers of mutations or incorrect copying. So to protect the organism, mutated cells that cannot be repaired often die. If the arrangement of amino acids in a polypeptide is different than it should be, the overall shape and folding of the protein will be affected. So we remember shape, so structure, determines function. Now we know the function will be affected. If the shape is different, the protein will no longer be able to perform the job it is supposed to. So the effect of mutations on gene expression. Changes in the DNA base sequence in DNA affect the activity of the cell by changing the proteins that are formed by the cell. So we have silent, nonsense, and missense. So no mutation means there's no genetic issue. Silent mutations mean they cannot be noticed. So we might change one base, but it doesn't change the amino acid coded. Nonsense means that we coded from a protein that has a function to a stop codon to stop the copying of that gene. And missense, we have conservative and non-conservative. Therefore, we're going to see a mistake in the DNA, which we will see the wrong amino acid incorporated into a protein, so made into a protein. If mutated body cells, which remember those are diploids, survive, we could see cancer as a result. If mutated sex cells, which is haploids, survive, the mutation can be passed on to the offspring. There are 3,000 genetic diseases that have been attributed to a mutation in just one base pair. One type of harmful mutation is progeria. It's premature aging. So the mutation in LMNA gene, a protein that holds the cell nucleus together. So it's death by early teens from heart attack or stroke. And the mutation is found in the sperm or egg cell, not necessarily in families. So another type of genetic mutation, we have Brooke Greenberg, 20 years old, did not develop physically or mentally. What type of syndrome could it have been? So some mutations have actually been beneficial to organisms. They can cause polypeptides to form that will increase an organism's chance for survival. So some examples are resistance to HIV and resistance to the plague. Now, genetic mutations and recombinations from sexual reproduction provide the raw materials for evolution. Another beneficial mutation example, we have normal white blood cells and a mutant white blood cell. The mutant white blood cell does not have a T cell with CCR5. And this example allows for people to survive the plague. Then we have another beneficial mutation, partially beneficial, so extramuscular. Now, this is an organism who is missing myostatin protein, which is supposed to slow muscle protein growth. Now, there are different types of mutations. Make sure you understand each of the different mutations we're going to cover. So the first type of mutation we're going to discuss is called a point mutation. It only affects a single base or codon. So substitution, which is one base is replaced or switched. And another type of point mutation is inversion, which is a group of bases becomes reversed within a DNA sequence. There's frame shift mutations where changes the entire reading frame. These two types, we have insertion where one or more bases are added to a DNA sequence. We also have deletion where one or more bases are lost from a DNA sequence. So frame shift mutations are dangerous because they are, when we see bases read as a triplet code, groups of 3. Remember, if we add a base or subtract a base, the total number has changed. So this changes the entire triplet code, so the entire codon. Everything that is read after could essentially be incorrect. So here are the different types of mutations. So deletion or frame shift, where one or more bases is lost. Then we have inversion, which is at a specific point, where groups of bases break off and becomes attached in a different order than it originally was. We have addition and substitution. Addition is where we add a base. Substitution is where one base replaces another. So addition is a type of frame shift mutation, and substitution is a type of point mutation. So the universal genetic code chart tells us what specific amino acids pair with our codons from mRNA. So let's look at an example. So after addition, so we have DNA after addition, we're going to see the correct base pairing. However, the polypeptide sequence we'll see is going to change. DNA sequence after deletion. So we're going to see the mRNA properly have the complementary bases, and the polypeptide sequence again will slightly change. DNA after substitution. So now from our original DNA sequence, we've substituted the G. So our amino acid sequence is going to change because of the complementary bases. Therefore, the polypeptide will change. So sickle cell anemia, a disease caused by substitution. What is sickle cell anemia? It's a genetic disorder that prevents normal hemoglobin production. So hemoglobin is blood. Hemoglobin is a protein in red blood cells that carries oxygen. Over 300 codons, which is 900 nucleotides, code for hemoglobin molecules. Sickle cell anemia results from the substitution of one nitrogenous base for another. So we have our partial amino acid sequence. If you look where we have the second set of codons, there is a mutation. So 900 are correct in the first normal red blood cell, but 899 are correct, which causes sickle cell anemia. Now there's types of chromosomal mutations. The first, well, what is a chromosomal mutation? It's changes that occur on the larger scale. So it's going to happen on the chromosome, not in the amino acid sequence necessarily. So there's duplication, where there's extra copies of genes that are made. There's deletion, where some of the genetic material of the chromosome breaks off. Inversion, when a broken chromosome segment gets inverted or turned the opposite way and put back onto the chromosome. Translocation, when a fragment from one chromosome breaks off and attaches to another chromosome. We actually see this in Down's syndrome. Now your teacher is going to give you a test yourself. Please use these slides to help you answer the questions, and of course go back and review them and ask your teacher if you have any questions.